All right, guys, we're going to talk through chapter 4 today, Gnathosome fishes, the most diverse group of vertebrates in the world. So we know that the two living groups of jawed fish, chondrichthyes, cartilaginous fish, and osteichthyes, or bony fish, comprise the most diverse group of vertebrates with about 31,000 species throughout the world. The evolution of these guys really had two major developments. One, the development of a hinged jaw from the most anterior pair of primitive pharyngeal arches. That actually was one of the most important evolutionary milestones for all of vertebrate evolution. Jaws permitted the capture and ingestion of a much wider array of foods. They allowed for defensive purposes, both interspecific and intraspecific combat. And the term gnathosome includes both jawed fishes and all tetrapods that came after the fish. And the second major development was the evolution of paired appendages. So early fish became much more active. As they became more active, they would have experienced instability while in motion, and most likely this favored appendages that would help them resist pitch and yaw. So those forces that they'd want to counteract. This led to the evolution of the first paired fins. Paired fins really help add stability to a streamlined body. The pectoral fins that project out from the sides were used for balancing and turning, and the pelvic fins really served more as stabilizers. Some of the fins projecting horizontally near the anterior of the body are there to help counteract pitch, while yaw is controlled by the vertical fins, like those coming off of the dorsal, such as the dorsal fin. So two of the earliest groups that we see, the one that we commonly call spiny sharks, appeared about 440 million years ago. This group of vertebrates were mostly small fish. The majority were less than about 20 centimeters long. They had large eyes and small nostrils. The internal skeleton was composed partly of bone and also a well-developed lateral line system. We talked through lateral line systems in our last lecture. They were active swimming fish. They were adapted to open water. Sometimes they've been included in the other group of placoderms. And although they survived into the lower Permian, they never were a dominant group, and they were really overshadowed by placoderms. So placoderms also possessed jaws, and their bodies were covered with dermal bony plates. They became the dominant fish during the Devonian period. These guys also possessed an internal skeleton of bone and cartilage and sort of this sharp dermal armor around the jaws. They can see some of the examples on the right side here. Relatively flattened bodies of some of these species point to the fact that they were bottom dwellers. They developed two distinct body segments that you can see in that picture on the right as well. And that was articulated with a ball and socket joint near the head. So this made the head really for the first time freely movable up and down. It allowed for a wider range of vision and gape. And we also see really the first record of live bearing birth with this group of fish, with documenting viviparity for the first time in vertebrates. So the placoderms evolved about 380 million years ago. And as I mentioned, both internal fertilization and viviparity originated first with this group. So the chondrichthyes, let's talk a little more about the extant groups that we see with chondrichthyes. This is the class that includes sharks and skates and rays. This group of fish are really distinguished by the primarily cartilaginous skeleton and by placoid scales with a posteriorly projecting spine of dentin. The cartilaginous skeleton represents a secondary loss of bone in this group of species. So they would have come from bony ancestors and reverted back to cartilaginous skeletons. By the Cenozoic era, chondrichthyes were present in large numbers and had greatly diversified. Another distinguishing factor of this group is that males have claspers on their pelvic area, which is a specialization associated with internal fertilization. Skates and rays are in their own superorder. Skates differ from rays in that they have a much more muscular tail. They usually have 2 dorsal fins and sometimes a caudal fin. They lay eggs rather than live birth. Skates and rays differ from sharks by having large pectoral fins that attach up around the head, no anal fins, horizontal gill openings, and eyes and spiracles located on the top of their heads. Bony fish. So, Osteichthyes are the largest group of living fish. They've been a dominant form of aquatic life for the last 180 million years. They comprise about 97% of all species of fish out there. And there are 2 major groups that we'll talk through. The lobe-fin fish and the ray-fin fish. So the lobe-fin fish, and we've got an example of one of these in the photo, possess a really interesting muscular lobe-paired appendages. It's supported by an internal skeleton. Each of these groups are treated separately because it does appear that they have been on their own independent evolutionary journey for a long time. These guys are the only living animals with a functional intracranial joint and paired appendages. And their appendages are coordinated in a way muscularly and with the nervous system that's much more like tetrapod limbs. Now, the rest of our lecture really focuses in more on the ray-fin fish. So as we talk through the rest of this fish lecture, that will be our focus. So I'm going to talk through the systems of our different groups of fish, and as we did with our jawless fish. And it gets pretty interesting with these guys. So skin. Unlike most vertebrates, most fish have an epidermis that consists really entirely of living cells. They have glands that produce mucus. Sometimes those glands also allow for toxic secretions and other substances. The glands are more abundant in fish that lack scales. The dermis in most fish is characterized by the presence of scales. and those scales are composed of bony and fibrous materials. The book goes into a lot of detail about the 5 different types of scales that we see, the different type of fish that we see the scales occurring on. So take the time to read through that. Fish that either lack scales entirely or have a reduced number of scales tend to be bottom dwellers or in moving water, fish that hang out in caves, fish that are in crevices or other tight spaces. Sometimes they're fast-moving pelagic fish like swordfish or mackerel. So the loss of scales, of course, increases flexibility and decreases drag or friction on the fish. So some of these fish really secondarily lost scales as an adaptive strategy for the niche they occupy. Some of those fish that lost scales actually have internalized scales embedded in a secondary skin covering. Of course, we see coloration in some fish skin and scales. Coloration's produced by all sorts of different pigments and serves several different purposes. Color change in some fish is controlled by the nervous and endocrine system. It can be brought about either by changing the shape of chromatophores or by redistributing pigment in the chromatophores. So we see multicellular epidermal glands of at least 42 families of fish that are also modified to function as light-emitting organs known as photophores. Photophores are fascinating, and I have a brief video on the next slide you can link to learn more about some of our deep water dwellers that use light emitting as part of their strategy. So most what we would call luminous fish, those that emit light, live in a depth of 300 to about 1,000 meters. Some of these fish that live at those depths do move up vertically towards surface waters at night for what we would call a feeding migration, a daily feeding migration. Some luminous fish produce light chemically by interaction of an enzyme with a phenol. Other species, like the one in the video that I linked, that live in deep waters have a symbiotic relationship with bioluminescent bacteria that reside in specialized glands or pouches that the fish hold as a structure. And because that bacteria glows constantly, those fish have to have a method for covering it up. And they can do that in a number of ways. But we know that luminous fish use those light for interspecific communication. They use it for camouflage, or they use it for attracting food. I want to talk just a bit about the skeletal system in bony fish. The book describes really in detail the composition of the skull and the vertebral column. It's worth noting that a greatly constricted notochord that runs through the center of each centrum of the spinal column, it really fills the spaces between adjacent vertebrae. So the vertebral column actually extends from the skull to the tip of the tail in most fish, and fish vertebrae are differentiated into trunk vertebrae and also into caudal vertebrae. The development of stabilizing fins that we mentioned earlier is found in both the bony and the cartilaginous fish. The pectoral girdle braces the anterior pair of pectoral fins. In most fish, these fins are operating not just as stabilizers, but also as diving planes. They're set at angles that help generate lift for the fish, the anterior part of the body. And in some species, that's particularly important. The unpaired median fins as we mentioned, assists with stabilizing the body, and that includes the dorsal fins, the anal fin, and the caudal fin. Here's the link to just a brief video that is sort of dramatic, but tells you a little bit about this really interesting lanternfish of the deep seas. Now, I'm actually going to end the lecture there and start with recording the next section in a bit.